So good morning everyone. Today me and Paywen is doing revision on the immunity chapters, and in this chapter we have divided into 5 parts, which is phagocytes, immune response, antibody, monoclonal antibody, and vaccination. So I will be covering phagocytes, immune response, antibody, and monoclonal antibody, and vaccination will be covered by Paywen later. So we will start off with doing revisions on the phagocytes. And from what we have learned, we actually have learned 2 types of phagocyte, which is neutrophil and monocytes. And neutrophil is actually short-lived, whereas for the monocytes is actually long-lived. So this means that after neutrophil phagocytose, it will actually die and produce a thing called pus. And this pus consists of the dead white blood cell, dead bacteria, tissue fluid, and also the damaged tissue. So we can see here is a diagram that shows us that pus form under the skin. And for monocytes, it's actually long-lived, so it will not die after phagocytose. So the actions of phagocytosis is actually start off with the chemotaxis, which is released by the bacteria, and this will attract the phagocytes, which is either neutrophil or monocytes, to come to move towards the chemical, and it will recognize and attach to the antigens of the on the surface of the bacterium, and after that, the phagocytes will actually undergo a process called endocytosis or phagocytosis, and the bacteria will within the phagocytotic vacuole will actually digest by the hydrolytic enzyme, which is secreted by the lysozyme, and fusion of lysosome with phagocytotic vacuole to form a phagolysosome or secondary lysosome. And the last step for the phagocytosis is actually killing and digestion. The killing process is actually done by the hydrogen peroxide and toxic free radicals, whereas the digestion is actually done by the enzyme, which is, for example, protease can break the bond of the in the bacteria there, so that it can also kill the bacteria. So first question, macrophages synthesize intracellular enzyme. Figure 2.1 is a summary diagram of events that occurs in the macrophages. Name the process at A. So process A, I'll open this to the floor. Anyone? Yes, Jung-Un is endocytosis. So actually, we can also say phagocytosis, but pinocytosis here is rejectable, is because pinocytosis is actually self-drinking. And engulfing also ignore, because engulfing is actually an action, is not a process. Second question, name the stages of protein synthesis that occurs at E and F. So E is actually nucleus, and F here is ribosome. So what is the process in E and F? Anyone wants to try? Yes, Jung-Un, you got it. So for E is actually transcription, and for F is actually translation. So name the organelles B, G, H, and J. So, sorry, B, G, H, and J. Anyone want to try? No? Rough endoplasmic reticulum. Yes, Andrea, you are right. Oh, sorry. G is actually G is actually ribosomes because it pointed in the dots there, so it's ribosomes. And H Golgi apparatus, yes. J is mitochondrion, yes. And so B you can say is actually the you can say phagocytotic or endocytotic vacuole or phagosome, and you cannot say it's a vacuole or vesicles or Golgi vesicles or excretory vesicles, and it ignores food or pathogenic. And then for G is actually can accept rough endoplasmic reticulum, sorry. So but it ignores the 70S ribosomes is because this is only found in the prokaryotics and not eukaryotic cell. And H is Golgi body apparatus or complex. And then for J is mitochondrion or mitochondria.
Sian, can we go back to G? G, no, just now the answer, mark scheme G. You see, at the end there, reject RER and rough ER. So that means when the student is being asked to name the structure, looks like you have to give the full name and spell out the name instead of using the RER, which is an official short form, is not allowed. So that means you need to learn up the full name and the spelling. Okay. And another thing is,
thank you, teacher. So describe what happens to the bacteria between C and D. So C and D is actually the phagocytosis process, and the bacteria are actually destroyed or broken down, or you can say hydrolyzed, and by the hydrolytic enzyme. For example, you can also give an example for carbohydrates, lysozyme, protease, and nuclease. So I've mentioned just now that the enzyme is actually used to digest the bacteria. So, and the bacteria is actually killed by the hydrogen peroxides or free radicals. So you can include this in your answer as well. Then if you want, you also can name the substrate of the enzymes. For example, you can say polypeptides, nucleic acid, lipids, and you can name the bonds that has been broken. So maybe peptide bond is broken by the enzyme protease, and phosphodiester bond is broken by the enzyme nucleic acid nuclease. Sorry.
What about the glycosidic bond and the ester?
Glycosidic bond is broken by the enzyme carbohydrates, and the ester is actually the lipase. And then for phagocytes and lymphocytes are part of body cellular response to infection by pathogen. Figure 6.1 shows the origins and maturations of phagocytes and lymphocytes. So here is a picture show that this is actually bone marrow, and there are 3 different type of the white blood cells being released, and this is macrophages, B lymphocytes, and T lymphocytes. So, and neutrophil here. So how we identify macrophages is actually has 2 characteristics. So wants to try what's the 2 characteristics that we can use to identify the macrophages? Yes, Yong-eun is indented nucleus, and another one. Agranulocytes. Yeah. We can also say it's eccentrically placed nucleus. And then for neutrophil, we actually have the multi-lobe nuclei. Yes. So for B lymphocytes, we can identify by, you can see that the nucleus is almost covering the whole cell. So this is actually lymphocytes. But here, how we identify it's a B lymphocytes or T lymphocytes is because you can see that from the bone marrow, the B lymphocytes is directly like released into our blood. However, for the T lymphocytes, it actually will pass through an organ called thymus, and in thymus, actually the T lymphocytes will mature there and only will release into our blood. So it's actually it's actually T lymphocytes.
Okay,
So
can I add a little bit here, Sian? Now this part is not in the syllabus, the maturation of B cells and T cells. It was in the old syllabus, but it is not in the current new syllabus. Okay, so but since you have touched on it, so I'll just mention about maturation. Maturation of B cells and T cells is when the cell receptors are formed. So B cells have got the antibody receptors form, and the T cells got a TCR, T cell receptor form. So for B cells, it will mature in our body is in the bone marrow, so it's B cell. For T cell, it will mature in thymus gland in D, so therefore it's called T cell. T comes from the word thymus. So when they mature is when all the T cell receptors for T cell and also antibody receptors for B cells will be formed. Okay, if it happen to be carrying the receptors that can recognize our self antigen, so those B cells and T cells carrying receptor that can recognize our own antigen will be removed so that it will not be released into our circulation. Okay, so that is maturation part. Thank you.
Thank you, teacher. So I'll use this question to do a recap on the whole immune response process. So explain the role of the cell A, B, and C in the immune response. So A is actually macrophages, B is B lymphocytes, and C is T lymphocytes. In your answer, use the terms antigen and non-self. So for the definition for antigen is actually any large molecule recognized by the body as a non-self or foreign can stimulates our immune response. So since the questions ask to write our answer in antigen and non-self, so if you write the whole definitions of antigens, you can get 2 marks because here they mention antigens are non-self and their foreign antigens will actually induce our immune response. So first, the first step of the immune response is actually the macrophages phagocytose the bacteria and then cut up the bacteria into small pieces and present it on the cell surface membrane of the macrophages. So this actually is the first step of the immune response. And then after that, the B cell or T cell will recognize these antigens presenting on by the macrophages, and the specific B cell or T cell have the complementary receptors with these antigens will actually come and bind to the antigens, and this will lead to the next stage, which is clonal expansion of the B cell or T cell. You can also say that it can divide by mitosis. And after that, after the clonal expansion, they will actually form two types of cell, which is memory cell and active B or T lymphocyte cell. And another thing is you can actually, from here, is actually you can zoom into to talk about the T helper cell, T cytotoxic cell or T killer cell, and B cell. So for T helper cell, it actually secretes the cytokinase to stimulate the B cell, and the cytokinase will also stimulate the macrophages to undergo the phagocytosis towards the pathogen. And for T cell or T killer cell, it will actually destroy the pathogen and by producing the perforin. So like just now, I've mentioned that the T killer cell is actually the bacteria is actually killed by the free radicals or the hydrogen peroxide. And for the B cells is actually will become plasma cell, and it will actually secretes the antibodies to bind to the antigens and to phagocytose by the macrophages. So they also accept the answers macrophages, non-specific and faster response, and refer to specificity of the B cell and T cell, and also the antibody variable region in the is the antigen binding site. So this antigen variable region I will discuss it later. So here is the whole process of the immune response in our body. So figure 2.2 shows part of the immune response to the first infection by a bacterial pathogen that has entered the body through the lining of a bronchiole. J and K are stages in the immune response. So this is the process that I've discussed just now. So state what is happening at stage J. Anyone wants to try? No.
Sian, I will suggest you to call any one of them since it's very dormant.
Okay, then I will call Sarah. Do you want to try?
Is J phagocytosis?
Yes, you're right. So J is actually phagocytosis, endocytosis, or you can describe in terms of engulfing or forming phagosome. And then explain the role of cell L at stage K in the immune response. So cell L is actually macrophages, and response K is actually the antigen presenting. So you can say that the digestions of the bacteria to destroy the bacteria of pathogens so that to prevent it spread through the body, and antigen presentations, or you can say the bacteria is presented on the cell surface membrane of the macrophages.
No, antigen presented, not bacteria.
Oh, sorry, antigen presented on the cell surface membrane of the macrophages. And you can say the specific B cell and T cell is undergo the clonal selection, and you can also say recognitions binding of the appropriate B cell or T cell. So macrophages are antigen presenting cell APCs. Antigen from pathogens such as the bacteria shown in figure 2.1 are presented to helper T lymphocytes as shown in figure 2.2. Very few helper T lymphocytes respond to the presence of APC by binding in the way shown in figure 2.2. So you can see here the helper T lymphocytes actually have the receptors which can bind to the antigens presented by the antigen presenting cell. So you can say that only few numbers of
Hang on. What is the question?
With very few T lymphocytes respond to the presence of APC by binding the ratio. Oh, sorry.
Can you move the mark scheme down so we can read the question for C?
Wait, I think. Oh, suggest why this is so.
Oh, suggest why this is so. Okay, thanks.
So you can say that the idea that there is only small number of the specific T lymphocytes will actually have this correct specificity, and because different T lymphocytes are specific to different antigens, and the T cell receptors is complementary in shape to the antigen. So you can also mention about this maybe during a primary immune response, so no memory cells. But I think this answer is not so strong to support support this question. So now,
just continue from this answer, right? So that means it's trying to tell us that actually in the primary immune response, during the antigen presentation, there are actually very few number of T helper cells as well as the B cells that will be activated, because we've got so many different types of clones of T helper and B cells. So therefore, the first activation means clonal selection. Clonal selection means the T helper and B cells that can bind to antigen, the numbers are very small. Because of that, after clonal selection, it must be followed by clonal expansion. That is when we increase the number of those T helper cells and B cells that are specific against the antigen. I think because of that, it's trying to tell us that the initial clonal selection is a small number of cells that are being activated. Thank you, Sian.
Okay, thank you, teacher. So now I will doing some recap on the antibody structure. So from this picture, we can see that the antibody structure is actually has a hinge region, and this region is actually to provide the flexibility for the antigen to bind to the for the antibody to bind to the antigen, which is differently spaced. So, and the antibodies actually consist of the four polypeptide chain, which is 2 light chain and 2 heavy chain. So for the light chain and heavy chain, they actually join by the disulfide bridges. You can see here. For the light chain with the heavy chain, they actually join with only 1 disulfide bridges. But if between the 2 heavy polypeptide chain, it's actually joined with 2 disulfide bridges. And for these two, we actually it is known as the antigen binding site, which can bind to the antigen on the cell surface of the bacteria. So there is also we also know this region as a FAB region, which is the fragment antigen binding, and this region as FC region, which is a fragment crystallizable. So, and we also have this 2 chain of sugar molecules and different like for the animals and human antibodies, the positions of the change of sugar molecules is actually different. So during an immune response, plasma cells secrete antibody molecules. Figure 1.1 is a diagram of an antibody molecules. The diagram is not complete. So draw a circle around a variable region. I will draw it. So the variable region is actually, you can see from this diagram, the variable region is actually this 2. So we can, this 2 is the antigen, is the variable region. And draw in and label the position of the disulfide bonds in the molecule. So like what I've mentioned just now, between the heavy and light chain, they actually only have 1 disulfide bridges, and between the 2 heavy polypeptide chains, we actually have 2 disulfide bridges. So is something like this. And explain the importance of disulfide bonds in the protein structures such as antibodies. So the disulfide bonds are between the cysteine residue, or you can say between the R groups. Is actually you can also say that it's a covalent bond. The disulfide bridges is actually a covalent bond, and it is a strong bond and not easily broken. And this disulfide bridges actually will hold the polypeptide chain together in the protein with tertiary or quaternary structures, so that the protein can maintain its shape, or it can maintain its 3D structures, or you can say that it can stop loss of shape or prevent deforming.
So Sian, so this question is actually based on chapter 2, which is biological molecule, right? So the question that they ask you in one question can overlap between different or among different chapters. So this is a basic of the cross links between the cysteine R group in protein tertiary and quaternary structure. Yeah, thank you.
Thank you, teacher. So describe how antibodies provide protection against pathogen. So you can see that actually I've prepared some points here, and actually these points got some mistake, and it's actually some of the points are invalid. So I want you guys to type your answers. Like I will give you guys 1 minute to read through the questions, and you type the number that you feel that is invalid, or it will not get any marks from in this question. Yeah.
So that means choose a question that's not valid lah. And choose the answer that's not valid.
Yeah. Actually, there are only 4 valid points here.
Wow. Okay. Thanks.
Two, 4, 5, 6, and 9. Yes, Andrea, but there is one of the point you stated is actually correct. So so you
Okay. Start.
So the correct point is actually 1, 6, 7. 8 is actually got half correct, half wrong. And if we I think Jung-eun got all the points. Yeah, I think Jung-eun
2
got all the points.
4 5 8 9 more.
Oh yeah, you left out 3, sorry.
So can you
So,
explain, please?
Because from this mark scheme, the first point is actually secrete by the plasma cell. So antibodies secrete by the plasma cell. So it is one of the answers here. And number 2 is wrong, is because antibody bind to the diseases. So it's actually not binds to the disease, it's actually bind to the antigen. So it's reject disease and it's combines with the antigen. And the point 3 is refer to the specificity of the antigens. This point you can include in your answers. And now the point 3 is antibody has complementary binding sites with the antigens is actually it's actually not stated in the mark scheme. And for point number 4, there are receptors on antibody. So it's actually the variable regions is antigen binding regions, and receptors of antibodies actually rejected. And you can also state for the function of antibodies, because antibodies actually will carry out you will actually neutralize the toxin. And for the point number so for point number 6, you can say the antigens will actually lyse the pathogen, but you cannot say that it's break down the pathogen. For number 7, you can say prevent viruses and entering the cells. And for number 8, you can state that the bacteria is agglutinate or clump the bacteria together, but you cannot say it's coagulate. So for point number 8 here, coagulate is actually wrong, because the antibody can only clump the bacteria together, they will not coagulate.
So
And
coagulate means solidify lah.
Yeah.
Whereas clump together means clump together. So they are all still soluble. And cannot be a bacteria is not soluble, right? But you cannot see them lah. Okay.
And then for point number 9, you can say it's opsonization or enable recognition by the phagocytes so that they can phagocytose the bacteria. So for point number 9 is invalid, because phagocytes kill the bacteria. You actually need to specify how the phagocytes kill the bacteria is by phagocytosis. And for point number 10, you can say the codes of bacteria to facilitate the phagocytosis. So this is interrelated to point number 8 and point number 9. And point number 11 is actually receptors on phagocytes for constant regions of antibodies. So for point number 8 here, enable recognitions by the phagocytes, you can get 1 mark from this answer, because it's mentioned in the mark scheme here.
Wait, I disagree with your answer. I disagree with point number 1. If I mark their answer, I will not give mark because it's irrelevant to the question. It's way out from what the question is asking. How antibody provide protection? Who cares who is producing? That one is out, because how many marks is this question? 4 marks. How can I give so many marks for a 4 marks question? So number 1 is totally out of the question. Please cancel off number 1.
Okay.
Definitely, I will not give mark because it's not what the question is asking for. Okay, then that means you're supposed to tell the examiner the 6 ways how antibody acts on the antigen. So here, can you see my sharing or not? So these are the 6 ways that you need to mention. And then you need to use back the actual word that is being used to describe the process. Like just now, Sian said agglutination rather than coagulation. So you cannot simply use the word. You must use back the word that they use, right? Prevent entry, prevent movement. How come no prevent movement? Agglutination, lysis. Lysis is not the antibody that causes lysis. You see properly, it's not the antibody that causes lysis. The antibody activates lysis because it activates another molecule to punch hole and some other chemicals enter the cell. Okay, so therefore the cell lysis occurs because punch hole by another molecule, not by antibody. So I will not say antibody causes lysis. I'll say antibody activate the lysis. So I'm very, very particular about the accuracy of the description in your sentence one. If you say antibody causes lysis, it means antibody is the one that causes lysis. No, it is not antibody. It is another substance activated by this antibody. Okay, so write your answer as accurate as possible because all of you want to score A star, right? A star and top in Malaysia and top in the world, right? So don't compare with those at average. You're above average, okay? So right answer there is professional. Opsonization. Opsonization just now, you see the mark scheme, Sian, can you show your mark scheme, please? And the last one is neutralization, right? Okay. Opsonization, please show that mark scheme. I think today, Pei Wen no need to present. Can you show the mark scheme, please? You see, point number 9, opsonization or opsonin is the name of the antibody, right? A means accept, enable recognition. So what they're trying to say here is that the opsonization of the bacteria, that means the coating of the bacteria with the antibody, enable the bacteria to be recognized by the phagocytes because phagocytes contain receptor for the FC region of the antibody. So that's the meaning of this accept, enable recognition. Opsonization add the markers onto the bacteria, and these markers are able to be recognized by the phagocytes, which are carrying receptor for the FC region of antibody. So you need to understand the mark scheme very clearly. Okay. All right. Thank you, Sian. Pass to you.
So the last question is, other proteins are found in cell surface membrane. Describe 3 roles of the proteins in the cell surface membrane. So actually this is chapter 4, but so the first point we can write is the channel protein or carrier protein is for facilitated diffusion.
Okay, stop. This is not in this chapter, so we pass it to Pei Wen. Thank you.
Okay.
Because of short of time. Okay, sorry. Yes, speak and clear. Thank you.
Since there's no much questions about vaccination in the past year paper, so I'll just go through it with the points here. Vaccination is an artificial active immunity. It is when foreign antigens are introduced into the body artificially, either by injection or oral consumption. It is to activate the first immune response in order to produce memory cells to give us the long-term immunity. So in the subsequent exposure to the same antigen, the memory cell will activate secondary immune response. So there will be a faster and higher antibody level production and infection without symptoms, reservoir, and transmission. These 3 are the main purpose of vaccines. And vaccine is a preparation containing antigens that come in many forms, for example, whole life microorganism or kill microorganism. And then the problems with vaccines, they are only for infectious disease. It means that the immune system will act against foreign antigens, for example, virus, bacteria, and protozoa. And the second one, live versus that bacteria or viruses. Live vaccine is more effective because living microorganisms can multiply in the body, so antigen is continuously being produced, so there will be more memory cells. While for that bacteria or virus, APC, the antigen presenting cell cannot present antigens from that virus. Therefore, immune system cannot be activated. For a poor response happens when the person is born with defective immune system or protein energy malnutrition, so they cannot produce cell for immune system. Antigenic variation. The antigens encountered by our immune system keep changing because there are many different strains but can cause the same disease and regular mutations. Antigenic concealment. Foreign antigens are hidden away from immune system. For example, intracellular pathogen covering their body with host antigens and antigen in external environment in the body. The last one will be safety or side effects. So in order to make vaccination successful, we have to vaccinate nearly everyone in a population, which is herd immunity. So there will be no symptoms, no reservoir, and no transmission. Number 2, continuous vaccination, same, no symptoms, no reservoir, no transmission, and no recurrent. For monoclonal antibody, it is an immunoglobulin produced by a single clone of plasma cell. The clone of plasma cells are genetically identical, produced by repeated mitotic division from one parental B cell upon exposure to specific antigen. Monoclonal antibody molecules are identical, so they can only bind to one specific antigen. Cell line is a cell structure developed from a single cell, and therefore the cells are genetically identical. So, monoclonal antibody is produced by hybridoma method in the lab. Hybridoma is a cell formed from the fusion of two different types of cells, which are B cells and cancer cells. This cancer cell divide indefinitely, and plasma cell originate from B cell and undergo differentiation. They secrete antibodies. So when the membrane of these two cells fuse together, a heterokaryon is formed. And when the nuclei of these two cells fuse together, a hybridoma is formed. The hybridoma is then cultured in the lab and divided by mitosis. This clone of hybridoma will secrete monoclonal antibodies for diagnosis and treatment. So these are the steps. First one, immunization. Antigen will be injected into the mouse so that it will activate the immune system to produce lots of plasma cell against the antigen. Antibodies secreted by the plasma cell will have similar shape with the antibody receptor of the cell. And then after 2 to 3 weeks, the mouse will be killed, and then the spleen will be removed from the mouse and flush out the plasma cells. The plasma cells then will combine with myeloma cells, for example, which are the cancer cells of bone marrow. The cell will be transferred into into each wells of microtiter plate. Then we need to select and culture the hybridoma cells. So the cells will divide into many cells and secrete antibodies. These antibodies will be purified for diagnosis and treatment. So monoclonal antibody can be used for diagnosis, for example, blood typing before transfusion, tissue typing before transplant, locating blood clot in the body, identifying, locating cancer cells, identification of pathogens, distinguishing between different strains of certain pathogens and different types of leukemia, and for pregnancy test. So suggest the advantage of using monoclonal antibodies in diagnosis of disease. So first, we know that monoclonal antibodies are specific for antigen. So we have to state that monoclonal antibodies are highly specific for the protein or other antigen that they detect by binding. As hybridoma can be cultured continuously, there's an unlimited supply for monoclonal antibody, which makes them readily available. These are the answer scheme. They also say that this monoclonal antibody can distinguish between different pathogens, and they can be labeled and tagged, and then detect location of tissue expressing antigen, and then faster diagnosis can detect antibody levels. Okay, outline the use of monoclonal antibodies in the treatment of disease. Anyone want to try? No?
You can talk about cancer.
Maybe I'll just explain because it's running out of time. Okay, let's look at the answer scheme. So first we state that each type of monoclonal antibody will target a specific targeted antigen in the body, and then they bind to the receptors or antigens on cell surface of the diseased cell. They kill the cell by stimulating the immune system. And then the extra point will be attach radiative substance or drug to kill or treat the diseased cell. And you can give example like treatment of Ebola, rabies, or tetanus. Okay, I think that's all from me.
Okay. So for the last question, diseased cell, using monoclonal antibodies to target the diseased cells in our body, that means targeting our human cell, either it is infected by the virus or bacteria that enters our own cell, or it is a cancer cell. Is that what it means? Treatment of disease, target treatment to specific disease cell means actually targeting to our human cell. Right? Right, targeting our human cell, either the cell already contains the virus or bacteria inside it, and hence is presenting the cell outside there, or it is a cancer cell. So it will contain a non-self antigen. So then the antibody come and bind to this cell so that it will promote phagocytosis by the phagocytes, or you can attach radioactive chemicals onto this antibody to actually kill the cancer cell, or you can attach drugs to this antibody so that this drug will be sent to this deceased cell only and target the disease cell. So this is called magic bullet, right? Magic bullet, targeting the medicine towards that particular particular disease cell. I think that's what they're trying to say here, treat disease, right? And also recognition of antigens on pathogens, so can also target pathogens directly, which is very interesting. Recognition of antigen on pathogens. Ignore bacteria unless stated that they cause disease. Yeah, okay, be very careful. Our body got a lot of bacteria, fungus, virus, but not all of them cause disease. So only the monoclonal antibody is used is only to target that bacteria that cause disease, which is the pathogen. Okay? All right. Okay, is it that's all, Pei Wen?
Yes, teacher.
Yeah, thank you so much, Pei Wen. I always like your presentation because it's so crystal clear.
Thank you, teacher.
Yeah.